So next is about how root nodule is formed in biological nitrogen fixation. Especially in leguminous plants only this root nodule is happening, right? So this is a legume like plant, isn't it? It is having a marginal placentation, a pod is there. Okay, this is a pea plant, and on the root you can see a like hemoglobin pigment, that rhizobium bacteria is the one who is participating. All these basic knowledge and all we all know, but how this nodule is forming, how this what this root system is bearing a nodule, that you should understand. All these bacteria name are all everybody knows it. So but first of all, when we talk about this relationship is called as symbiotic relationship, right? Which is a symbiont type. The host cell and the symbiotic relationship is happening here. A symbiont has been created. Okay. So how we are going to form this root nodule? So on the surface, on the leaf surface or root surface, you can see that there are some the plant is being you can see some receptors for plant surface. There are some receptors. So plant surface, especially the root surface, because root inside the root only nodules are formed, right? So this receptors are for lectins. Okay, this receptors will take lectins. What are lectins then? Lectins are the glycoproteins. They are carbohydrate rich molecules, right? So on the root surface there is receptors for binding of this lectins, and this is the root hair, right? On this root hair surface there is a receptors for binding of lectins. So lectins will directly interact with the glycoproteins of the bacteria. Okay, bacterium is there. So bacterium is the one who is making this root nodule, right? Multiplication of rhizobium bacteria. Okay, so this this glycoprotein of the bacterium and also the lectins get interacted. So there is a host symbiont specificity is there. Host symbiont specificity is there. So this rhizobium bacteria is showing a good association for the leguminous plant. So first point you should remember is the lectins, which helps in creating a specificity, especially the glycoprotein of the bacterial surface is being interacted with the receptor of the plant root hair. Okay, so lectins is a key proteins, glycoproteins that is involved in rhizobium legume symbiotic association. Sometimes the calcium is also playing an important role. Okay, calcium also, the divalent cations are also playing an important role in this thing. So there is an attacking of this bacteria has to attack this particular root surface, right? So root will release some amino acids like tryptophan. Hope you know that tryptophan is the precursor molecule for our auxins. So auxins helps in IAA, indole triacetic acid. So these auxins are helping in promoting the growth of the root. So now this rhizobium has to give a point of attachment on the root hair. So there is a chemical thing that has been surrounding in this area. First we know that it is a lectin. Next it should have some chemoattractants, just like amino acid, sugars and organic acids. So all these are some of the chemoattractants. Here it is given that flavonoids. Flavonoid is a secondary metabolite, right? They also helps in providing the chemoattractant substances, chemical stimulants. Okay, so this is this chemo attraction has been done by our flavonoids, amino acids and some sugary organic acids. Okay, there this what the rhizobia bacteria will attack to the root surface. That is some of the theories. Okay, so some of the supporting theories for rhizobium to attack this root hair is using chemo attraction method. And now this bacteria will evade this root hairs, and what happens, this is that root surface is called as rhizosphere. Okay, and because of all these signal molecules there is a curling of, here you can see that curling of curling. That means the root hair has been curled and enclosing this rhizobia bacteria. Okay, and this curling, we want some node genes. Okay, and nodulation factors. Node genes is also called as nodulation factors. So some node genes has to be there in order to curl this root hairs. Okay, sometimes chitin is also helps in the nodule formation. Okay, curling of this hair. Okay, now there is a twisted, distorted type of root you can see here. Okay, and this node factors will helps for it can also increase the root hair formation, so according to some theory. Now you can see that the rhizobium is completely enclosed inside the root curling. Okay, now what is happening? The root hair will completely encircle the rhizobium. Now this rhizobium has to enter into the root hair, into the epidermis, epiblem, and then cortical region. So here the plasma, a cell wall has to be dissociated, right? So some signal molecules are there, and the signal molecules will help in the dissolving of this membrane of our plant cells, root, especially root area, and this light pink color should that bacteria has to move along. So just like ants are moving, the rhizobium is moving in a depending upon the what signal molecule they are moving. If you have a signal just like a traffic, every car will be moving in a single line. Just like that, ants also moving in a single line, because of there is a chemostatic attraction, there is a signal that has been happening. So you can see that this bacterium will form an infection thread. This point is very important. This rhizobium bacteria is forming a infection thread, a line, a series of line. Okay, and it enters into the root cortical area. Especially they are evading the meristematic regions. Okay, meristematic regions. So it is actively dividing. So you can see from primary nodule center to secondary nodule center, they move. They move along the pericycle region. Okay, now in due course of time, this they undergo division, right? This bacteria has to undergo division, right? And you can see that this after infection thread, it enters into the cortical region, and then it reaches the pericycle, it undergo active division, and in second stage there is a large number of rhizobia will enter into the large number of rhizobia bacteria will enter into this, penetrate into the host wall and get into the in between the cell wall and plasma membrane. Everything they have to enter, no, right? They have to dissolve all this membrane, cell wall, cellulose, everything has to be, they have to enter, and it forms a this is called, this thread like infection is called as infection thread. So there is a tubular intrusion of this bacteria into the cell, thereby now we have reached here. Okay, now it has to enter the cortical region, and all the final stage, this rhizobium will infect all the cells, cortical cells, and what you can see that the bacteria start dividing. It undergo multiplication, right? And this it cluster together and forms a bacterioids. Okay, it forms a bacterioids. It forms a nitrogen fixing cells. It can evade the particular area, cortical area, and it can evade, and it can encircle into a big size, and this is called as bacterioids. Okay, now they are with the peribacterioid membrane. The surrounding is called as peribacterioid membrane. So they cluster into a globule like thing, right? So there is a repeated division. The surrounding cell will undergo repeated division because here pressure has been created. So you can see that this root nodule has been formed. This bacterioids is being converted to a root nodule like structure. So this is almost pink in color, and that pigment is called as leg hemoglobin. Leguminous plant producing that pigment. So it is a red hemoglobin. So this determines the nitrogen capacity of the plant. So there is a symbiotic relationship of this rhizobia with the host cortical cells. Okay, so this is how the process is happening. So this is a root nodule. Here first point you should remember is the lectins, flavonoids, and node factors. Node factors are the nodulating genes. Okay, that that has to be interacted with the root surface. Okay, root, especially the plant root surface is bearing receptors for receiving the receiving the bacteria, rhizobium bacteria. So there is a lectin is there. Lectin is a glycoprotein. The chemoattractant is there. Then only the bacteria will come and sit here, isn't it? And for that there is a regulatory genes are there, and this node factors help in nodule formation. So curling of leaf occurs and it encapsulates the rhizobia. So after enclosing, the cell wall and plasma membrane will dissociate, and the rhizobium will enter into the root hair, just like an infection thread, just like a line infection thread, and it enters through the cortical cell, reaches the pericycle, and maximum development is seen in the cortical cell, and they undergo division, they enlarge in size, and they have a membrane called as peribacterial membrane, and this full structure is called as bacterioids, and this bacterioids surrounding cells will undergo division, and bacteria will undergo further multiplication, and bacterial stimulates the surrounding cell to undergo division to form a nodule. Okay, to form a nodule like outer covering, and thus you get a nodule formation. So this is an overall biological nitrogen fixation for leguminous plants. Okay, leguminous plant, how the root nodule is active, but inside that there is a lot of chemistry that is happening. This nitrogen fixation is created basically by dinitrogenase enzyme that we have already dealt previous classes. That is, it is having a iron protein and a molybdenum iron protein section subunits. Okay, and ATP is being broken down, that I have already told here. So dinitrogenase enzyme helps in the conversion of water, dinitrogen into the ammonia. So dinitrogenase enzyme is required for this conversion. So we were talking about both dinitrogen complex formation, and here we have learned about the biological nitrogen fixation routing using this root nodule formation. Hope it is clear.